a 1-minute video which is recorded in our lab. So this says something about the biological semen analysis lab, the accuracy of which has not been replicated anywhere in the world. I mean to say, this is the love tunnel, this is the fallopian tube, this is the oviduct, whatever the name which we give. But these identify. For example, if you see this particular spermatozoa, and I have used the incubator here, the unselected spermatozoa, which is a mixture of somewhat tyrosine phosphorylated, some abnormalities, everything. This particular oviduct is to repel or to propel that particular spermatozoa, which is recognized as ineligible, away from the oviduct. This is the sperm reservoir establishment model we have mimicked. Of course, this is buffalo spermatozoa. If you see all the good quality spermatozoa with high mitochondrial membrane potential, all green, and with no tyrosine phosphorylation or no capacitation in the head, they are only allowed to make a contact and establish a reservoir. Whereas these sort of abnormal or immotile spermatozoa or some other inherent quality which is not desired by that particular semen analysis lab, they are rejected. That means to say each and every spermatozoa which is being attached to the oviductal explant or oviductal reservoir is being subjected to thoroughly for a selection criteria. These fellows are trying to attach, but they are not allowed to attach. They are being moved away. So this video, this 1-minute video conveys a lot to us. See this fellow, he is phosphorylated, but he is not allowed to enter into this. So the significance of this video in this particular talk and its relevance to the topic, I'll come little later. So now I will get into my presentation. So this is the topic which we will be discussing for next 40 to 50 minutes. I will try to restrict the timing. So it is always a desire, or I can say even a dream of mankind to have or to produce offspring of desired sex. Not only in case of their own, in case of human beings, but also the animals which they own. They want a particular type of sex of offspring to be produced more. And it has remained as a dream. It has not been fully materialized with 100%, but there are some efforts, and that effort leads to 90% it is possible now. But the main thing is, geographically, although, for example, if you take a Western countries, then there is a demand for male offspring. It is more because of the meat production and other things. Whereas in our condition, in case of India, if you see, we go for female, especially in case of bovines, we want more male. Geographically, there is a variation in the desire for the sex, but the desire for having the sex of the offspring or pre-selection of the sex of the offspring is being there and has been long continued to be there. And here, that aroused a lot of interest among the researchers. And 100 years, especially after 1900, lot many researchers have been conducted in this particular area to understand what determines the sex of the offspring. So sex determination is the one thing which has been dealt very in detail, and it is now very clearly understood how it is being done. In case of mammals, it is chromosomal. In case of lower and other things, it is environmental. That is understood. But what we are going to talk is having understanding the sex differentiation, sex fixing, and sex ratio adjustment. Invariably, at sometimes, if you see, they are interchangeably used, but they are not actually similar. So there are minute differences between these three. But what we will be talking mainly is sex fixing and sex ratio adjustment. Whenever I discuss this topic with my students or with my colleagues, I ask this question: who fixed the sex? And invariably say because it is the male who produces X, Y spermatozoa, and it is his responsibility to fix the offspring of the sex. Coming to the next, see this situation. For start, that it is a chance because male delivers in millions of spermatozoa, and among those spermatozoa, one is getting fertilized. So it is a chance factor, and that's how it determines the sex. But you see these conditions. For example, I am inviting you to my home and I offer a cup of coffee, a cup of tea, both. So you select to drink either a cup of coffee or cup of tea. That is your preference. Whereas the person who gives is preferring giving equal amount, both X and Y. Similar is the case here. The choice. It is no more considered as a chance. Rather, it is the choice of the mother. That is called the cryptic female choice. But the mechanism of which is yet to be understood fully. Of course, partially, we do know that how she selects. I will come a little later about that. And this is one proof for that. If you take this, many of you might be already knowing the Chinese calendar. It takes only 2 factors. One is the age of the female at the time of conception and the month of conception. It gives almost 90% accuracy. That's what it is being said. So by using that sex determination concept, how we can select or pre-select the sex of the offspring, especially in case of bovines, because Indian context, bovine is very much important. But I'll take the help of the publications in human as well as mice, because not many studies have been conducted in that particular aspect. Coming to the Indian context, why? Many of you might be knowing, just to recapitulate the things. So you do very well. We have a huge livestock population. So coming to around 300 million bovines, means major milk producing animals. But what the data says is only 50% of those animals are really productive. Remaining 50% is, one cannot see the burden, but they are not exactly for the milk production purpose. People usually say that India is milk producing, it is topmost country because of large population. It is not so. There are technology based milk production enhancement is happening, especially in the last 2 decades. But in more number of animals is also a obviously reason, one reason. Third one is large number of unproductive animals. You know, the calving interval is even 2 years or 3 years under field conditions. So lot many unproductive animals, and the bulls important. We need almost around 8,000 bulls to cater the needs of the frozen salmon production in our country or requirement in the country. But we are not able to get it because ultimately the high genetic merit as well as high fertile bulls. Even if you take the bulls that are in the demonstration, not all are high fertile or medium fertile, some are low fertile as well. So ultimately what we say is we have a good number of animals, but majority of them are low productive or low performance animals. And especially if you take some of these crossbreds and other things, there will be a lot of stray cattle problem. And ultimately in the last 19th as well as 20th census, it is found that almost 5 million cattle are stray cattle. So that means to say, yes, now the time has come because the resource is shrinking, water is sinking, soil fertility is sinking, and then fodder cultivation is sinking. Everything is sinking, but the animal population remains intact. So it is a high time to think over and to come with less number of the animals producing more so that effectively we can utilize the resources. For that purpose, in case of males, if you want to have high genetic merit, then the one thing is sex pre-selection. This is going to give you. In the other female sex, high producing animals, we have to replicate in a faster way. So that's how this sex pre-selection is very important in that category, where it is important. Many of you may not be agreeing with me when I say this priority species is crossbred cattle. But I'm not telling this is not required in indigenous or buffaloes, but immediate priority is in crossbred, because in case of indigenous animals, the males can be used in the drought, as well as the indigenous cattle almost contributes around 20% of total milk production. But buffaloes, it is triple purpose, so not even a single animal is wasted. But here comes the problem, crossbred cattle. A major portion of the animals which are there, males is no man property and they create a lot of problems. So here, very immediately, we need to have some technology to produce more amount of females when compared to this. Having said so, how to go about? It is always better to revisit history, because it is always told that the person who do not know history is likely to repeat it. So that's how, as I told you earlier, sex determination has been a dream and has been interest of research for quite a long period. What they could understand is environmental influence on sex determination is more in case of fish and reptiles, whereas in case of birds and mammals, it is genetic. And as I told you earlier, 335 BCE, Aristotle, he proposed the first thought, yes, it is the male partner who determines the sex of the offspring, but it is the dominance. I'll come to a little later how dominance theory works out. So if the heat of the male is more at the time of intercourse, or the cold of the female is more, then automatically it results into male. And till 1900, this concept was believed. And after 1900, intense researches have been conducted and it identified the Y chromosome that controls the sex determination. And here comes the major milestone, that is in 1991, McLaren and Koopman et al. They have identified the sex determinating region for SRY. And many of you know that what is the contribution of SRY. It has a TDF, and that TDF acts on the cytolysyl and produces AMH, and that AMH arrests the Mullerian hormone, and that's how it develops into stress on that side. So ultimately, it has been concluded that yes, now it is the Y. Similarly, in case of DMRT gene is there, gene is there. This gene is responsible for sex determination in case of birth. So many of the factors that determines the sex of the offspring has been conclusively identified. And having said so, how to model it? So I'll take a little bit about this. This is full of human beings. This slide is full of human beings. And they try to modulate based on the having identified this is X, this is Y bearing spermatozoa. What are the differences between these two? It is characterized as the X chromosome is big and bit is slower, and it can stay more period in the female reproductive tract when compared to Y. And the challenges, it can face many challenges which are imposed by the female reproductive tract from the spermatozoa and cope up, but Y cannot. So comparatively, Y spermatozoa are thin, weaker, and have less DNA. DNA part I will come little later. So having said this, so based on these criteria, how one can separate these two or how one can skew sex ratio. These are all the 6 methods that has been proposed, and that was claimed as 75 to 80% accuracy in the literature. First is timing of sex, because this is important. That's how I'm explaining. Timing of sex, it is a Shuttlesbury and Billings method. It is always said X spermatozoa is having capacity to live in the female reproductive tract even in the adverse conditions for a long period when compared to Y-bearing spermatozoa. So, at the time of ovulation or after ovulation, if mating takes place, then there is every possibility that the sex of the fetus will be male. And even many of you agree, or many of friends in the field conditions, the veterinarians, they say when we go for late insemination, we get a male, more male calf in case of bovine. So this may be a reason for that. Second is sexual position. Of course, it is only exclusively for human being, but not for the animal. What it says is deeper depositions that results into the male, because in human being, the anterior part of the vagina is 2 to 5.5 pH. So this pH is very much sensitive, and majority of the spermatozoa cannot survive. So even if the deeper penetration, as I told you earlier, X, Y are weak. So if they are exposed to these acidic conditions, then they will be gone. So that's how it will be resulting in the female. So that's how when you go for deep penetration, surpassing that particular area, so there is a chance for male. And then sexual abstinence, when the sexual abstinence of the male is there for 4 or 5 days, and after that the intercourse takes place, then the forthcoming proportion will be more of Y, and that helps the male. And here comes the female aspect of changing the diet, more potassium, and even they have advocated meat, banana, apricot. If one takes more potassium containing food items at the time of ovulation, that is 14 days after menstrual period, and then there is a chance of getting male. And magnesium, these are all, I'm not telling 100% proved, but they have been proposed as a theory and with 75 to 80% accuracy. Timing of orgasm, I will not explain much. And the same, based on these concepts, yes, acidic genital environment favors female, alkaline genetic environment favors male. So Dooch method also was practiced to control the sex of the offspring. So ultimately, the one-line theme which I want to give all these things is there are differences which are perceptible between X and Y upon exposure to the female reproductive condition, and the pH or the environment, alkaline or acidic, that plays a major role in case of sex-free selection. Having said this, what are the more other accurate selections that we can employ for this sex-free selection? Obviously, spermatozoa, because there is X and Y. So we go for sex sorting, and then with sex-free semen directly A or produce embryo. And sexing of the embryos, we can go ahead with invasive or non-invasive method. I'm not going to explain the method, it will take a lot of time. So, but these methods, embryo sexing is not fully advocated because there is every possibility that certain damage takes place to the embryo, and it needs a lot of skill and other things.